Despite its appearance, this is not a UFO. It is a virus, and viruses are very interesting. For one, these are obligate intracellular parasites. In other words, they cannot survive without using a cell to replicate themselves. And viruses are often in this gray zone as whether or not they're alive or dead. And most people feel that they're probably not alive because they can't grow, they can't replicate on their own, they don't have independent metabolism, etc. And they have no cellular structure. But nonetheless, the important features of a virus for the MCAT exam is that it is composed of a head and a tail. The head part has a protein capsule called the capsid, and within this protein capsid is the genetic material, which may be DNA or RNA as genetic material, which is extremely unusual. The tail part is structured such that it can attach to a cell. And so a virus that is able to attach to a bacterium and infect bacteria is called a phage or a bacteriophage. As we know from personal experience, viruses can attack people as well, as you know from having a cold at some point in your life. And so when a virus attacks a cell, it will land on the cell, it will inject its genetic material into the cell, and it will force the cell to replicate more viruses. If sometimes the cell will replicate so many viruses that it will burst and release these viruses. That would be called a virulent state. Now, the virus may have two aspects to the life cycle. It may have a life cycle in which it infects and bursts cells and releases more viruses. That would be called either the lytic life cycle, lytic meaning to burst or tear apart, or it might be called the virulent life cycle. In the life cycle in which the virus remains dormant and quiet, that would be called the lysogenic life cycle. When you get into medical school, you will study microbiology in much greater detail. The microbiology for the MCAT exam is very basic. Microbiology is essentially the study of bugs, viruses, bacteria, and fungi. And so bacteria are considered to be prokaryotes, whereas fungi are eukaryotes. Prokaryotes being pre-nucleus, karyote referring to nucleus. Eukaryotes being true nucleus, literally. And so eukaryotes include fungi, but it also includes all animal cells and plant cells, etc. Commonly on the MCAT exam, they specifically try to determine whether or not you know the difference between prokaryotes and eukaryotes. And so they would tend to ask questions to delineate that difference. So I've written a number of things here: nucleus, cell wall, ribosomes, organelles, and plasma membrane. I would like, based on your knowledge, you can pause here and you can see if you remember if these particular things represent a similarity between eukaryotes and prokaryotes, or a difference between, meaning that they're present in one but not the other. So when we look at nucleus, clearly there is no nucleus in a prokaryote. That's why they're pre-nucleus. And there are nuclei present in all eukaryotes, true nucleus. For the cell wall, you might have thought that that's a difference between the two, because you may have remembered that bacteria have cell walls. And that is indeed true. Bacteria do have cell walls, but some eukaryotes have cell walls. Like fungi, they have cell walls. And so it's not a difference, but rather this is a similarity. So nucleus is a difference, cell wall is a similarity. Ribosomes is a similarity. Both bacteria and eukaryotes do have ribosomes that helps to produce the proteins which we saw earlier. Then there are organelles. Prokaryotes have no nuclei and no organelles. So they have no mitochondria, they have no endoplasmic reticulum, etc. But they do produce energy. They can produce ATP like eukaryotes do, but they don't produce them using a mitochondria. Rather, they have their own internal membranes in order to produce ATP. In terms of plasma membrane, indeed, this is a similarity. Both bacteria and eukaryotes have plasma membranes. We already saw how the plasma membrane develops and is produced and exists in eukaryotes. And for the prokaryotes, they have a cell wall, but they indeed also have a plasma membrane associated with the cell wall. So they have both. So this is actually a similarity between the two. We will now look at bioenergetics. Bioenergetics is the study that looks at how biological organisms, like our cells, produce and use energy. We're going to begin the story at the end, ATP. ATP is adenosine triphosphate. ATP is the #1 short-term energy storage molecule. This is the molecule that we will break down to produce energy so that we can exercise, do sports, sit, read, learn the MCAT exam, whatever it may be. ATP is broken down into ADP, adenosine diphosphate, and an inorganic phosphate molecule. It is that reaction, the breaking down of ATP, that permits us to have the energy necessary to do biological processes. Now, there are many different mechanisms in order to produce ATP. And there are many different sources in order to go through mechanisms to produce ATP. The most common source to produce ATP is glucose. Of course, glucose is a carbohydrate. It is stored in our liver as glycogen. It is also stored in muscle cells as glycogen as well. It is broken down, it floats through our bloodstream, and then it goes to our cells. And then it actually will enter our cells, and that's when we will use it. But there are other molecules that we can use as well. For example, lipids. Lipids can be broken down from fat, and then the lipids can be sent into the Krebs cycle, where the energy from the lipids will produce ATP. There are also amino acids. Amino acids are broken down from proteins, and the amino acids can be fed into the Krebs cycle as well in order to produce energy. Now, the first source of energy that we use is glucose. If we start running out of that, then we will start using the lipids. And of course, lipids is stored in our body as fat. And then when that is broken down and we still require energy, that's when we start going after the protein. And protein is stored in our body as muscle. Now, glucose is our #1 source of energy, and then we look at the different mechanisms in order to produce ATP. The first mechanism is to break down glucose, and that is called glycolysis. Now, you may recall that lysis is a word that is used often in biological processes to infer that something is broken apart. So glycolysis is the breaking down of glucose. We just call it glycolysis. Now, the first step in glycolysis is that once the glucose enters the cell, and we'll see later that that process is helped by insulin, When the glucose is in the cell, the first thing that occurs in glycolysis is that a phosphate group is put on the glucose molecule. There are two reasons for this. To begin with, phosphate is a large, relatively large, negatively charged group. By putting this on the glucose molecule, it will prevent glucose from diffusing out of the cell. Because you may remember that when you have a charged molecule, it is less likely that it will cross the plasma membrane because of the lipid interior. So that's one good reason to put a phosphate group on the glucose. The second good reason is because just by putting the phosphate group on the glucose, you've changed the glucose, you've altered it. And therefore, the concentration of glucose within the cell suddenly becomes much lower. And because you'll have more glucose outside of the cell, you've therefore increased the gradient for glucose to enter the cell, which is a good thing for the cell because the cell needs sources of energy. So then the first thing is glucose is phosphorylated. It has a phosphate group placed on it. So now we have glucose 6-phosphate. Then glucose will change its form and become fructose. Fructose and glucose are both 6-carbon sugars. And so the only thing that's going to change is the order and kinds of bonds. And therefore, these are isomers. Glucose 6-phosphate and fructose 6-phosphate are indeed isomers. Now, fructose will have another phosphate group placed on it. So it goes from fructose 6-phosphate to 1,6-diphosphate. Now, the interesting thing about this is that by having two phosphate groups on one molecule, these are two relatively large negatively charged groups that are attached to the one molecule. Well, this is quite unstable because, of course, like charges repel just as opposite charges attract. So by having these two large like charges that are repelling each other on the same molecule, this is going to be part and parcel of the fact that this molecule is going to break apart. So you have this 6-carbon sugar with two negatively charged groups, which will break apart into 3-carbon sugars, 2 of them. And that accounts for the 6 carbons. So now we will have 2 phosphate trioses. The word triose suggests there are 3 carbons, tri, and ose suggests that it's a sugar. Just like fructose and glucose are sugars, well, so are these phosphate trioses, which are produced as a result of the lysis of the glucose molecule. Then these trioses will go through a series of steps and then produce pyruvate. And pyruvate becomes the key step in these series of steps of glycolysis, because it's with pyruvate that the decision has to be made. Is there oxygen or is there not any oxygen? Because so far, the series of steps that we have done in glycolysis did not require oxygen. They are a series of steps that just occur in the cell's cytosol, in the solution in the cell. Now the decision has to be made, oxygen or not. If there is no oxygen, then we continue along glycolysis and the pyruvate will produce lactate or lactic acid. And then we end up with a net of 2 ATP. So for per molecule of glucose, we end up with 2 ATP, which is relatively very little energy. However, in order to get that 2 ATP, it occurs very quickly. The other possibility is that the pyruvate will be converted to acetyl coenzyme A, that is, if there is oxygen present, and then the acetyl coenzyme A will enter the Krebs cycle, which occurs within the mitochondria of the cell. So those are the two possibilities of the conditions. Either it is anaerobic, which means no oxygen, or it is aerobic, which means there is oxygen present. One quick note about the enzymes. There are 2 types of enzymes that you really need to be able to recognize. and those are kinases and phosphorylases. Those are simply enzymes that can add or subtract phosphate groups. And those are enzymes that you should be able to recognize. So that is one of the possible mechanisms, is glycolysis, which will produce 2 ATP per molecule of glucose. The other possible mechanism is that the pyruvate will send acetyl coenzyme A into the mitochondria, and the acetyl coenzyme A in the mitochondria will enter the Krebs cycle. The Krebs cycle is also called the tricarboxylic acid cycle. For every molecule of glucose, you get 2 turns around the Krebs cycle. There are 4 important products of the Krebs cycle. For one, there is carbon dioxide produced as a waste product of the Krebs cycle. Because it is a waste product, we need to get rid of it. And carbon dioxide is a molecule that we get rid of in the lungs. We just blow it off in the lungs all the time. And that is a waste product of the tricarboxylic acid cycle, or the Krebs cycle. The second important product that comes out of the Krebs cycle is GTP. GTP is a molecule similar to ATP. And in fact, GTP, for every 1 molecule of GTP, it is equivalent to 1 molecule of ATP. Another important product of the Krebs cycle is NADH plus H. NADH plus H is considered to be reducing equivalents. They are sometimes called hydrogens. They are also sometimes simply called electrons. But NADH plus H is an important product coming from the Krebs cycle. And the fourth product is FADH2. And again, FADH2 is sometimes called reducing equivalents or hydrogens. So these are the 4 important products. It is interesting to note that ATP is not directly produced from the Krebs cycle. After the Krebs cycle, we go to the electron transport chain. And all these things, the Krebs cycle and electron transport chain, are associated with the matrix of the mitochondria and the inner membranes of the mitochondria. Remember, the mitochondria is considered to be the powerhouse of the cell. This is the most efficient use of glucose in order to produce the maximum number of ATP molecules. So now we get in the electron transport chain a series of events in which the hydrogens from FADH2 and NADH plus H go through a series of steps, and the final step is that oxygen receives the hydrogens, producing water as the final waste product of the Krebs cycle. So water, H2O, being a waste product just like carbon dioxide, is usually released in the kidneys, but there are a series of other ways that we can get rid of water. So this is the way that energy is produced. Now it so happens that for every 1 molecule of NADH plus H, you end up producing 3 ATP. For every 1 molecule of FADH2, you end up producing 2 ATP. So when you calculate all of this, you end up that if you take glucose as your source of energy and it goes through the Krebs cycle, an electron transport chain, in other words, you take glucose and it goes through aerobic respiration, you end up with a total of 38 ATP per molecule of glucose. Your net ATP will be 36 because it will cost you 2 ATP in order to get the acetyl coenzyme A into the mitochondria. So then you get a total of 36 ATP per molecule of glucose during aerobic respiration, but you only get 2 ATP per molecule through anaerobic respiration. Now there is something called oxygen debt. That is that you run to class because you're a little bit late, which of course is very rare for you, But nonetheless, you run to class, you sit down, and you're huffing and puffing. Well, that's quite odd, because sure, you were running and so on, but now you're sitting down, you're relaxed, you're content, and yet you're huffing and puffing. Now, why should you be doing that if you're not even doing any activity at that moment? Well, the reason is that when you were running, you needed energy quickly. And the body gets its energy quickly by using anaerobic respiration. It's much faster. It has fewer steps than going through the Krebs cycle, going around twice the Krebs cycle, and then in the inner membrane, going through the electron transport chain and all the steps involved there. That takes a lot of time. So though anaerobic respiration is not as efficient as aerobic respiration, which gets 36 ATP per molecule, it's much faster. So when you were running, you were able to use the anaerobic respiration, but you built up lactic acid. And building up lactic acid in muscles is not good. And if you build up too much of it, you can actually start getting cramps. So then you run, you get quick energy, you sit down, and then your body says, well, this lactic acid that you've built up, let's oxidize it back up to pyruvate, then we'll send it to acetyl coenzyme A, and then we'll send it through the Krebs cycle, and then that will be much more efficient than just sitting with our 2 ATP per molecule of glucose. So therefore, you sit down and you start breathing heavily as you're taking in extra oxygen in order to oxidize your lactic acid up to pyruvate, acetyl coenzyme A, and then going through the Krebs cycle, etc. So that is what oxygen debt is. The brain, the spinal cord, and the peripheral nervous system are composed of nerve tissue. The fundamental unit of nerve tissue is the neuron, and you're looking at a cartoon of the neuron here. I must mention, however, there are cells called glial cells, and these are supporting cells of neurons. The most important type of glial cell is called a Schwann cell. And we'll talk about Schwann cells in a few moments. To begin with, however, in terms of the anatomy of a neuron, we have here the cell body, which contains the nucleus. The cell body is also called the soma. Then we have different processes or fibers. The processes or fibers that tend to bring signals to the cell body, these are called the dendrites. The process or fiber that sends signals away from the cell body, that would be called the axon. When we go along the axon will end in little buttons. Because it terminates in little buttons, they're called terminal boutons. They are also called synaptic terminals. Now, the synaptic terminals have a little space after, and that's called the synapse. So in other words, between this cell and, say, for example, another cell that starts down here, there's going to be a little space, and that's called the synaptic cleft or the synapse. What happens is that when there is a signal going down the axon and then along here to the synaptic terminal, in the synaptic terminal there will be vesicles, little cellular containers of different types of substances. Some of these substances will be excitatory, other will be inhibitory, meaning it will inhibit the next cell or it will excite the next cell. An example of an excitatory substance is acetylcholine. An inhibitory substance is GABA. So these are chemicals that could be in vesicles that will fuse with the synaptic terminal and then diffuse across the synaptic cleft and then affect the next cell. This area, though it's called just a synaptic cleft, it has a special name when the next cell is muscle. And that special name is a motor end plate. So this is the general setup of the neuron. Now the neuron itself, as you may remember from physics in terms of discussing what capacitance is, the neuron has a separation of charges. The inner part of the neuron is relatively negatively charged, and the outer part of the neuron is relatively positively charged. So there is a membrane potential Vm, and the membrane potential is negative because the interior of the cell is relatively negatively charged. This is very important. To begin with, you could see that the plasma membrane is acting as a capacitor because it separates and stores charge. That is a definition of a capacitor. So there's a potential across the membrane. The reason why there is a difference in charge across the membrane is based on two concepts. To begin with, there's a sodium-potassium ATPase, which is the pump in the membrane. Now, the membrane of a neuron is called a plasmalemma or a neurolemma. It's the same thing as a plasma membrane. And so within the plasma membrane is this pump called the sodium-potassium ATPase. So it's an active pump. It uses ATP, it breaks it down, uses the energy in order to pump 3 sodium out and 2 potassium in. Now you may recall from general chemistry, sodium is 1 plus, potassium is 1 plus. So if you pump 3 sodium out and 2 potassium in, then you're getting a net 1 positive charge out. Therefore, if you're continually sending a net 1 positive charge out, therefore the outside of the membrane will be relatively positively charged and the inside of the membrane relatively negatively charged. Furthermore, because there's more sodium outside the membrane, more potassium inside of the membrane, and because the plasma membrane is relatively leaky to potassium, meaning that potassium will, going along its gradient, leak out of the cell, again, there is a relative movement of positive charges out of the cell, therefore leaving the inside of the cell negatively charged. Now that's quite important. Because of this resting potential of the cell, which is relatively negative, then what happens is that when the cell becomes stimulated or excited and sends a potential along the cell, which is an action potential, what happens is that the positive charges outside the membrane start to enter the cell, which means that gates open, proteins called gates open in the plasma membrane, allowing sodium to rush inside. And because sodium starts rushing inside, instead of the membrane potential being negative, suddenly it becomes positive because so much sodium, which is positively charged, rushes inside of the cell. This produces an action potential. And this potential will send a signal along the membrane. As positive charges start going inside, it sends a signal along the membrane, and then it ends up in the synaptic terminals or the terminal boutons, and then you can send chemicals diffusing across the cleft and then affecting the next cell. So then all these different neurons are arranged in our brain in such a way that they have different interactions with different groups of other cells. So this is the way the action potential will work along the membrane. Now there is something special that happens in peripheral nerves, and this has to do with those glial cells, which are the supporting cells. The cells called Schwann cells grow along the axons. And the Schwann cells can actually grow along the axon and then start wrapping themselves around and around. And so what happens is you end up getting these sheaths around the nerve cell or the neuron. And you get these sheaths which are caused by cells that are supporting the neuron that are just wrapping themselves and turning themselves around and around and around the axon. This helps to insulate the axon. Because it insulates the axon, it allows the action potential to move along the axon faster. Neurons and glial cells form the basis of the nervous system. The nervous system can be divided into the central nervous system and the peripheral nervous system. As we just saw in terms of the anatomy of a neuron, we saw that the cell body can also be called the soma. Well, if you have a collection of cell bodies in the central nervous system, that collection of cell bodies is also called a nucleus. A collection of cell bodies in the peripheral nervous system is also called a ganglion. Now, the central nervous system is essentially your brain and your spinal cord. The peripheral nervous system includes the nerves outside of the brain and spinal cord. Now, the peripheral nervous system can be further subdivided into the somatic nervous system and the autonomic nervous system. The truth is that the somatic nervous system and the autonomic nervous system anatomically also form the basis of the central nervous system as well as the peripheral nervous system. But classically, it's the peripheral nervous system is divided into the somatic nervous system and the autonomic nervous system. Now, the somatic nervous system is the nervous system over which you have control. To be more specific, it is innervated with nerves that go down to the skeletal muscles. So clearly, between those nerves and the skeletal muscles, you're going to have motor end plates. So you have control over your skeletal muscles. Those are the muscles that you use to walk and move and do all kinds of activities. So you have control over that. That is your somatic nervous system. The autonomic nervous system is the nervous system that innervates things that you don't really have control over. Things like your heart rate, the size of your pupillary muscles. These often tend to be more smooth muscles or cardiac muscles, but don't tend to be skeletal muscles. Now, the autonomic nervous system can in turn be divided into two systems: the sympathetic nervous system and the parasympathetic nervous system. Now, the sympathetic nervous system is the nervous system which is often called the fight, flight, or fright response nervous system. For example, as you're sitting there learning about the MCAT exam, imagine that a bunch of raptors walked into your room. And I'm not talking about the raptors that play for the Toronto team. I'm talking about real raptors from Steven Spielberg type movies. Now, if those kind of dinosaurs walked into your room, you probably will have certain effects coming from your nervous system that would be considered to be sympathetic nervous system effects. For example, what would happen to your heart rate? You would expect your heart rate would go up significantly because your heart's going to be pumping blood quickly in order to provide the oxygen necessary for your skeletal muscles so that you can run. And your pupillary size, what would happen to that? Well, clearly, they're going to increase in size. You can just think about Bugs Bunny when he's all excited or something's happening that's shocking or nerve-wracking. You see that the eyes pop out, and it's because your pupillary size will increase in order to get as much information as possible about what is going on. In other words, looking for possible exit routes or something of that sort. So in any kind of fight, flight, or fright responses, you can consider what would happen in terms of blood flow to certain parts of your body. Obviously, the blood flow to your gut will not increase. You don't need your gut at that point. You need blood flow to your skeletal muscles. You need energy to move or to be active, etc. So those are your sympathetic nervous system responses. Then there's the parasympathetic nervous system. That is considered to be your vegetative responses. So you're just lying down, reading a book, very relaxed, nothing too much is going on. So your heart rate tends to decrease. Your pupillary size tends to decrease because you do not need action or all the information necessary in order to run or jump or do something special. And the blood flow to your skeletal muscles will tend to decrease as the blood flow to things like your gut, more smooth muscle types of things, that can increase as you start to do some digesting. And people might say that, oh, I heard my stomach starts to grumble or whatever. And that is just, air that is passing along the gastrointestinal tract as you are beginning to digest and food, and is passing through the system, and blood supply is being given to the digestive tract. So this is the way the nervous system is divided. We will now examine the endocrine system. The endocrine system is the set of glands, tissues, and cells that secrete into body fluids. For the MCAT exam, essentially, the endocrine system comes down to the amount of tissues or organs that secrete hormones into the bloodstream. So hormones can be categorized into two main categories, either protein hormones or steroid hormones. Most of the hormones that we will be discussing will be protein hormones. So if I don't say anything specific, it means it's a protein. If I say something specific, then I will identify it as being a type of steroid hormone. Now, the effect that these hormones have on cells is quite different because, for example, protein hormones, because proteins are rather large and they're charged, they have great difficulty crossing the plasma membrane, as we've discussed previously. So protein hormones tend to have their effect by stimulating some sort of receptor on the plasma membrane. Then that receptor will start a domino effect. An important feature of this domino effect is a particular molecule within the cell which is called CAMP. That is cyclic AMP. Now you recall that ATP stands for adenosine triphosphate, so AMP is adenosine monophosphate. So cyclic AMP or CAMP is a very important mediator within the cell. So in other words, a protein hormone may stimulate a specific membrane receptor. That membrane receptor will go through a series of steps that will lead to increased concentration of cyclic AMP within the cell, and then this will result in a very specific interaction or effect that will eventually change protein concentrations or have some sort of effect on the cell, and so that the cell may affect other cells, etc. So that is how the protein hormone usually would work. Whereas the steroid hormone, and we know that steroid hormone has a certain structure which is fatty based on the cholesterol molecule. And so it has this fatty structure which can dissolve in the plasma membrane and therefore cross the plasma membrane. And so some steroids, because they can cross plasma membranes and cross the nuclear membrane, they can have an effect directly on the DNA. So that's a different mechanism of action than a protein hormone would have. Now, in terms of the axis, which means the levels at which the effect will occur from a hormone to the end organ, we have the hypothalamus, the pituitary, and the end organ that will be affected by the hormone. Now, it used to be thought that the pituitary was the master organ and that it controlled all the different events that were occurring in the endocrine system. But it has since been determined that the hypothalamus has many releasing factors, which means hormones released from the hypothalamus that will influence the pituitary gland and influence what types of hormones the pituitary gland will be releasing. Now, the effect of the hypothalamus and the pituitary gland functionally is part of the endocrine system, but anatomically, both these organs are in the brain, and therefore they're part of the central nervous system. Now, so the effect is the releasing factor going to the pituitary gland, having an effect, making the pituitary gland want to release particular hormones. The pituitary gland releases these hormones, they circulate in the bloodstream, then they go to a particular organ where it will have a particular function. When it hits that organ may, in response, start to produce another hormone. And when that other hormone produced by the organ starts to increase in concentration in the bloodstream, it will go back to the pituitary gland and say, slow down. So, and the reason is, because could you imagine if the pituitary gland just kept stimulating the organ to produce more and more hormone, then it would be no end. The organ would just always be producing more and more and more hormone, and the effect would just get out of control. So there is a feedback mechanism so that the pituitary gland knows that the organ is actually working, it's actually doing its job, and the pituitary gland can shut down its production of the hormone which is stimulating the organ. This feedback loop is called a negative feedback loop, which means the organ will produce the hormone, tell the pituitary gland to slow down. So this is one of the ways that an end organ can regulate secretion coming from the pituitary gland. This negative feedback loop sometimes includes the hypothalamus as well. So the pituitary gland secretes a bunch of different hormones. First of all, it is divided into two parts. There is a part of the pituitary gland towards the front that is anterior, and part towards the back which is posterior. The anterior pituitary gland secretes all these hormones. So let's examine each of these hormones. First, we have GH. GH stands for growth hormone. So clearly, growth hormone is important in, well, growth. And so growth hormone would have its greatest effect on bone and muscle, because you started off small and now you are where you are because growth hormone stimulated your bones and your muscles for you to grow. Next, there is TSH. TSH is thyroid stimulating hormone. So clearly, thyroid stimulating hormone goes to the thyroid gland and stimulates the thyroid gland. The thyroid gland is kind of interesting because the thyroid gland is located in your neck and it's shaped like a bow tie. So in the thyroid gland, there are a couple of hormones that are important, but the most important hormone secreted by the thyroid gland is thyroxine. Thyroxine is a hormone that contains 4 iodine atoms. And in fact, the thyroid gland is the part of your body with the highest concentration of iodine atoms. Thyroxine is secreted by the thyroid gland in order to increase your basal metabolic rate. So TSH coming from the anterior pituitary gland, going to the thyroid gland, stimulating it. Thyroxine is produced and your basal metabolic rate increases. So let's say you're a family doctor and you're in your office. Somebody comes into your office and they're all excited and hyper and all of that. Well, you might want to do some certain tests. You may want to run some drug studies. But one of the things that you may consider is doing thyroid function tests, because it might be that the person's thyroid gland is overactive and therefore they themselves are hyperactive. That's one of the things you might want to consider as thyroxine is being produced, stimulating the production of mitochondria, making more ATP being produced, and stimulating the metabolic rate of the person. On vice versa, you might have a patient come in who is very tired and exhausted and lots of fatigue all the time. And you may again want to do thyroid function tests to see if the person's thyroid gland is hypoactive or not producing enough thyroxine. Next on our list we have ACTH. ACTH is adrenocorticotropic hormone. So adrenocorticotropic hormone clearly would go to the adrenal gland. The adrenal gland is located on top of the kidney. So you have two kidneys, so you have two adrenal glands. They're rather small, and just like other parts of the body, there's an outer part called the cortex and an inner part called the medulla. We find this in many parts of the body. For example, the human brain has an outer part called the cortex and an inner part called the medulla. The kidney has an outer part called the cortex and an inner medulla. In fact, the word cortex originates from a Greek word which suggests bark. And of course, in a tree, there's the outer part, which is the cortex or bark. And it's similar for the adrenal gland, the kidney, the brain, and so on. So the adrenal gland has an outer part called the cortex, the inner part, the medulla. The medulla of the adrenal gland does not react to this axis. It reacts separately. In fact, the medulla of the adrenal gland is considered to be part of the sympathetic nervous system. The medulla of the adrenal gland secretes adrenaline, or what we call adrenaline. So it's coming from the adrenal gland, adrenaline. Some people call adrenaline epinephrine. There is a related hormone, noradrenaline. These hormones increase the fight, flight, or fright response. So the heart rate, respiratory rate, etc. The cortex of the adrenal gland is what is being stimulated by adrenocorticotropic hormone. Now, the adrenal cortex secretes three categories of important steroid hormones. It secretes glucocorticoids, mineralocorticoids, and the sex hormones. The sex hormones include male hormones such as testosterone and female hormones such as estrogen. The glucocorticoids, consider the word. It ends in oids, so you know it's a steroid. It starts with gluco, so if you had to think, what in the body would it increase or have a great effect on? It's going to be glucose. So glucocorticoids increase the concentration of glucose in the body. It's called glucocorticoids because it's the adrenal cortex, and examples of adrenal hormones that increase the concentration of glucose in the blood include cortisone and cortisol. So these increase the concentration of glucose in the blood. Then you have the mineralocorticoids, which increase the concentration of minerals in the blood, specifically sodium. Aldosterone is an example, a very important example, of a mineralocorticoid, increases the concentration of sodium in the blood. By doing so, by bringing sodium back into the bloodstream, water will come into the bloodstream by osmosis. So you increase the volume of fluid in the bloodstream, therefore increasing blood pressure. So you can imagine, in response to stress, blood loss, or fluid loss, such as a runner who's doing a lot of activities, you could imagine that ACTH is going to be increased coming from the anterior pituitary gland, stimulating the adrenal cortex in different ways, different effects on the different hormones, but essentially you end up getting more glucose liberated, and you get sodium being pulled back and water being pulled back into the bloodstream. So that's ACTH. Then there is prolactin. Lactin suggesting lactate. And to lactate is to provide milk. And so prolactin is a hormone important in breast development and milk production. In fact, I once had a patient in his 30s, presenting to the office, very shy as you can imagine, with a history of breast development and milk production. And in fact, he demonstrated to me that he was producing milk from his breasts, which were very developed. And so, without the help of a CT scan nor an MRI, you would be able to diagnose, as I did, that clearly there must be a tumor in the anterior pituitary gland, which is overproducing prolactin. And indeed, that was the case. The next hormone, FSH, which is follicle stimulating hormone. So in women, follicle stimulating hormone is secreted to stimulate follicles, and we will look at that in greater detail later. And in men, FSH is important in spermatogenesis. Some students like to remember the S in FSH standing for spermatogenesis. There's LH, which is luteinizing hormone, and we will look at its effect on the ovary and the menstrual cycle shortly. But in males, LH, some people like to remember that the L stands for Leydig, since the LH hormone is secreted by the anterior pituitary gland and stimulates Leydig cells in males, which in turn produces testosterone. So these are all the hormones of the anterior pituitary gland. The posterior pituitary gland is a little bit different. The reason is that the hypothalamus actually produces the hormones in the posterior pituitary gland. It produces it, releases it in the posterior pituitary gland, and then from the posterior pituitary gland, these hormones are released into general circulation. So the hormones coming from the posterior pituitary gland include oxytocin and vasopressin. Oxytocin has an effect on smooth muscle cells. It has an effect on smooth muscle cells of the breast to help contract them, which is important in milk letdown. It also has an effect on the uterus to help contract the uterus, which is important in delivery of the baby. Oxytocin is a hormone that we sometimes give to women when we do cesarean sections. Because when we do a cesarean section, after we remove the baby, then sometimes there could be a lot of bleeding of the uterus once we've opened the uterus to remove the baby. To help reduce the bleeding, giving oxytocin will contract the smooth muscle of the uterus, thus closing down many or most of the blood vessels. Then there is vasopressin, and the word suggests what it does. Vaso, referring to blood vessel, and pressin, referring to pressure. So vasopressin increases blood pressure. Vasopressin has another name, ADH, anti-diuretic hormone. Now, a diuretic is something that will increase urine flow. Anti-diuretic does the opposite. So ADH, or vasopressin, will bring fluid back into the body, thus reducing the amount of urine produced. So we're going to look at where ADH acts later. Special mention must be made of some hormones which are not part of the hypothalamus pituitary organ axis. For example, in the thyroid gland, there are a group of cells that secrete a hormone called calcitonin. Calcitonin decreases the concentration of calcium in the blood. Now, calcium regulation is intimately associated with cells that work on bone, because clearly in our body, the greatest deposit of calcium is going to be in our bone. So we have cells that create bone, osteoblasts. Clearly, if you create bone by putting calcium into the bone, you're going to decrease the concentration of calcium in the blood. So clearly, calcitonin would stimulate osteoblasts. And there are cells called osteoclasts. Osteoclasts are cells that will resorb bone, which means it will take apart bone, thus releasing calcium into the blood, therefore increasing the concentration of calcium in the blood. So calcitonin decreases the concentration. There are 4 glands that are around the thyroid gland. They are therefore called parathyroid glands. Now, when we operate on the thyroid gland, we have to be very careful to identify these parathyroid glands and preserve them. So these parathyroid glands secrete a hormone called parathyroid hormone. It's pretty easy to remember. It's also called parathormone or PTH. Now, parathyroid hormone, or PTH, has the opposite effect of calcitonin, meaning PTH will increase the concentration of calcium in the blood. Because it increases the concentration of calcium in the blood, it will act on osteoclasts. In the pancreas, we have several hormones that are being produced in a particular part of the pancreas called the islets of Langerhans. In the islets of Langerhans, there are cells that are called alpha cells and beta cells. Alpha cells contain a hormone, glucagon, which increases the concentration of glucose in the blood. And in your body, in fact, there are many hormones that increase the concentration of glucose in your blood. So if one of these hormones happen to be missing, you'll get away with it. You'll survive. You'll be just fine, in fact. However, there's only one hormone which will decrease the concentration of glucose in the blood. Just one. And because there's just one, if something happens to that hormone, because there's no redundancy in our system, then we end up with disease. And of course, this hormone, which is located in the beta cells in the islets of Langerhans, was discovered in the land of the Toronto Raptor, and this hormone is called insulin. So insulin is the hormone that will decrease the concentration of glucose in the blood. It has many effects, but one of its effects is to actually help glucose go from extracellular to intracellular. And as you already know, through glycolysis, once it gets in the cell, it is phosphorylated, and then it goes through the rigmarole. So insulin decreases the concentration of glucose in the blood.